Hello and welcome to this lesson for Advanced Higher Biology. This one is looking at Unit 1, Key Area 1. We're on laboratory techniques for biologists, and specifically we're looking at Key Area 1D, protein detection. So prior to this, we have looked at the separation techniques of both centrifugation and gel electrophoresis. In this lesson, we're going to look at how we can detect specific proteins if we have separated them out. And then we'll move on to look at how we can use microscopes in order to look at different types of tissue. So for today, we are looking for you to understand how immunological techniques are used to detect and identify proteins. You need to be able to describe what we mean by an immunoassay technique. You need to be able to name the type of antibodies and describe the chemical labels that are used in this technique. And finally, you need to be able to explain when the Western blotting technique would be used and describe the process that's involved in this. So to begin with a quick recap of what we're talking about when we refer to antibodies and antigens. Now, those of you who did higher human biology might be a little bit more familiar with this than those of you who did higher biology. But the basics are that an antibody is a Y-shaped globular protein, and that is normally produced as part of the immune response by the body. And our immune systems use these antibodies to bind specific antigens. Now, an antigen is normally the small molecule present on the outside of a bacterial or a viral cell. And the immune system produces antibodies specific to those antigens in order to weaken or to destroy the pathogen itself. And you may have heard some of these terms, particularly recently when we've been discussing COVID-19 in the news, etc. So an immunoassay technique essentially uses or exploits this ability of antibodies and antigens to bind to each other and to do so with specificity. It's similar to the process of an enzyme and its substrate binding, but in this case a reaction is not necessarily catalyzed, so as a result you're not going to lose either molecule. So when we're wanting to identify proteins that are either antigens or antibodies, an immunoassay technique is usually the type of process that you would use. Now, when they are formed naturally, an antibody will usually take on several different shapes. So that receptor site will bind to different parts of the antigen. So as you can see in this one here, if we've got our antigen, we've got several different Y-shaped antibody molecules that are shaped in order to bind on to different parts of our antigen. And this is a process which is referred to being as polyclonal. So we've got more than one type of cell. However, when we use them in the lab, we tend to just use monoclonal antibodies. And that just means it's an antibody that is all one type and they all have the same specificity and are going to bind to the same part of the antigen. So an immunoassay then is something which uses our monoclonal antibodies, and it's usually got some kind of reporter molecule attached. Now, often this is an enzyme, but it could also be some kind of chemiluminescence molecule or a fluorescence molecule. Essentially, the basic process occurs when the antigen is present in the sample. If we have that antigen, then adding the antibody to the same sample will allow the antibody to bind to the antigen. When that happens, the reporter molecule will set off its particular cascade. So that might be an enzyme catalyzing a colour change reaction. And that allows the investigator to detect the presence of the enzyme. If we see the colour change, we know that the enzyme has reacted, and therefore that tells us that the antigen is there, and therefore we've detected our protein. However, it will also let us quantify how much fluorescence or colour change you are seeing, and consequently exactly how much protein you have available, how much antigen there is. If the antigen is not present, then the antibody can't bind to anything, and as a result the enzyme will be washed away from the sample before any reaction is able to take place. So there'll be no colour change or fluorescence in this particular case. And that tells you that you do not have that antigen present. Now, the similar process may also be used when we want to use a specific antigen in this time to detect the presence of antibodies. For example, when we are doing COVID-19 antibody testing, so one of the things they have been looking at is how long these natural antibodies may last in the body after exposure to the virus. And what you'd be doing in this case is you'd be taking a viral protein which has got the antigens present on it. You're going to put that on some kind of solid support, usually some kind of gel matrix, and you're then going to flood this solid support with antibodies from the patient sample. So we're going to be looking at, in this case, it would be serum or blood that would be being used. So you've got your solid support with your COVID-19 viral protein present on it. And then you're going to flood this area with blood or serum from a patient who you suspect has had COVID-19, and we're looking to see whether or not they've got the antibodies present in their blood. If they do, normally we then have a secondary antibody that we're going to introduce into the sample, and this secondary antibody will be specific to the first, and it will have the detectable label attached. So we're looking at having three different components here. We're looking at having our viral protein, the antibody in the patient sample, and our secondary antibody, which has the detectable label attached. So with the viral protein being available, the antibody in the patient sample will bind onto that, and then the secondary antibody with the detectable label will attach. Consequently, that's when you would then see your protein color, your enzyme color change, or your fluorescence or your chemiluminescence, and that would tell you that the antibodies are present. If the antibody is not present, then it won't obviously bind to the viral protein, but not only that, it won't bind to the secondary antibody with a detectable label, so that won't show up and you won't be able to see the presence of that, and that would indicate that this individual had not come across that virus before and did not have the antibodies already present in their blood. Western blotting again exploits some of these natural techniques. So it's a technique that we use to identify specific proteins, and these are ones that have been separated using SDS page gel electrophoresis, which we discussed in the last lesson. So first of all, the first stage in the process is to allow these proteins to be separated on that gel. We then use several different processes in order to blot, this is where Western blotting gets its name, that those proteins from the gel onto a more solid medium. And essentially that's just more convenient for further study. It can be stored a little bit easier and managed a little bit easier rather than a more fragile gel. We're then going to stain the secondary medium, usually referred to as a filter. We're going to stain that with fluorescent labelled monoclonal antibodies or other reporters. So in the same way as we just have. So if we have got our protein bound onto our gel, we've got our primary antibody, our secondary antibody with our enzyme, and when they bind, the substrate is catalysed by the enzyme, and that gives us our colour change signal again. So it's usually fluorescence in this case, but you're going to see the enzyme catalysing at that and giving us that signal. And once those antibodies have bound to their target proteins, you would then wash off the extra. So after there's been an opportunity for these primary and secondary antibodies to bind to the protein and to each other, the filter would then be washed out, just leaving those ones behind. As this is on our formal gel, it's been blotted onto our more robust gel, we're then going to go through a visualization process where you would expose that gel to light of different wavelengths, and the fluorescent labelled antibodies would then be able to fluoresce, and that gives us the precise location of the specific target proteins, and that allows them to be identified. So from that, you should be able to describe what we mean by an amino acid technique, name and describe the antibodies and chemical labels used in this technique, and explain when the Western blotting technique would be used, and roughly describe that process. As ever, please get in touch if you do have any questions.